Hello everyone, welcome to ISF College of Pharmacy. As you know that ISF College of Pharmacy is conducting this ISFCP dialogue series under the Aegis of Internal Quality Assurance Cell. In this series, every day, eminent pharmaceutical scientists from pharmaceutical fraternity is coming to our platform and delivering a scientific talk. In this special segment under MOU activity with Smriti College of Pharmaceutical Sciences, Education and Research, we are conducting this lecture today. So, for this occasion, we have a very eminent speaker today with us. I welcome Dr. Kushagra Dubey, Madam, Professor in Scope College of Pharmacy. Madam has total 15 years of experience in academic and research, currently working as a professor, Department of Pharmaceutical Chemistry in Smriti College of Pharmaceutical Education, Indore. Ma'am is having a strong academic research background as she has published more than 3 dozens of research papers in national and international journal of repute, as well as she has edited many books, book chapters like 5 book chapters. One Indian patent has been also granted. Ma'am has attended more than 1 dozen short-term training programs as well as faculty development programs. In addition to this profiling, she has received more than 15 prestigious awards like Young Researcher Award, Best Faculty Award, Innovation Award for Scientific Research, and Excellent Teacher Award. She is also reviewer of many journal, international and at national level. She has developed the innovative teaching practices and assessment for the evaluation in teaching research as well as its co-curricular training. She is a registered pharmacist in the State Pharmacy Council Madhya Pradesh and is a lifetime member of Association of Pharmaceutical Teachers of India. She has coordinated number of sponsored academic, scientific and technical events. Overall, she is a good human being, a good researcher as well as a good teacher. With this bio sketch, I would like to please, ma'am, start.
Okay. Thank you very much, sir. Oh, I'm not sir able to see my. Uh, sir, 1 minute. Yeah. Sir, I'm audible
Yes, yes, ma'am, you are very audible.
and my screen is visible.
Yes, yes, it is visible, ma'am.
Oh, so thank you very much, sir. And I am very thankful to the coordinator of MOU activity ISFC and Scope Dialogue series, Dr. Saurabh sir, for giving me this opportunity to share my knowledge. Today, my topic of talk is drug repurposing, a novel drug approach for drug discovery and development. So, this is the overview of my topic, like introduction to drug repurposing versus traditional drug discovery, strategies of drug repurposing, approaches of drug repurposing, and method of drug repurposing, in which I am going to tell about the molecular docking studies. So, if we talk about the introduction of drug repurposing, then the drug repurposing have many synonyms like drug repositioning, drug retasking, drug reprofiling, drug rescuing, drug recycling, drug redirection, and therapeutic switching. And it is the process of identification of new pharmacological indication or the therapeutic indication from the old or existing or investigational or products or approved drugs or the drugs which are already in the market. And it is the application of the newly developed drugs for the treatment of disease other than the drugs originally intended for the therapeutic use. For example, if we have a drug which is approved for diabetes, then it is repurposed as an antihypertensive drug. So the drug's main pharmacological action is for the as an anti-diabetic agent, but now it is used for as an antihypertensive drug. And it also involves establishing new therapeutic use for already existing or known drugs. So This is all about the drug repurposing, and it has been accounted at approximately 30% of the US Food and Drug Administration approved drugs, and the vaccines or biologics are repositioned drugs. And the first example of drug repositioning was an accidentally discovery which was observed in 1920. And after that, more approaches were developed for accelerating the process of drug repositioning. And the most and the best common examples are the aspirin, sildenafil, minoxidil, methotrexate, etc. These are the very common examples. So, if we talk about the drug discovery, then there is a word traditional drug discovery. So, how it is related with the drug repositioning, or how it is different from the drug positioning or repurposing. So, the traditional approach to drug discovery, it involves the de novo identification, and it included the 5 stages. The first one is discovery and preclinical. Second is safety review. Third one is clinical review. Fourth is FDA review. And the last one is FDA post market safety monitoring. So these are the 5 main stages of the traditional drug discovery, but it is a time consuming, costly process and have a high risk of failure. On the other hand, if we talk about the drug repurposing, then drug repurposing or reprofiling consists of the 4 stages. First one is compound identification, second one is compound acquisition, third is development, and the fourth one is FDA post-market safety monitoring. So in this, with the advancement of bioinformatics, cheminformatics tools, and availability of huge biological and structural database, drug repositioning decrease the time and cost of the drug development with reduction in risk of failure. Like we are originating the new target of the old therapeutic indication, new target for the new therapeutic indication, and the old target for the new therapeutic indication. So the discovery of drugs by this approach is certainly advantageous over the traditional drug discovery. For example, if we talk about the sildenafil, it is a phosphodiesterase inhibitor, initially developed for the coronary artery disease by Pfizer in 1985, but it is now used for the treatment of erectile dysfunction. So it reduces the development cost and definitely the development time also. The second most best example is the metformin, which is an oral anti-diabetic drug used for type 2 diabetes mellitus, but now it is used in the cancer therapeutic, and it is under phase 2 and phase 3 clinical trials also. So when comparing with this traditional drug discovery with the drug repurposing, it is seen that in traditional approach, it is estimated that 10 to 16 years are spent for the development of the new drug. While if we talk about the drug repurposing, then the estimated time is between 3 and 12 years. Okay. If we talk in sense of the cost, then it only costs $1.6 billion to develop a new drug using drug repositioning strategy, while the drug development through traditional strategy costs around $12 billion. Okay. A reposition drug does not require the initial 6 to 9 years which is typically required for the development of the drug in traditional process. But instead, the drug is directly entered to preclinical testing and clinical trials so that it reduce the overall cost, time, and risk for the development of the drug. So this is the traditional drug discovery versus the drug repositioning. There were many examples of drug repurposing which are available. Like for example, rosiglitazone. It is approved as an anti-diabetic drug, but now it is repurposed as an anti-malarial drug. Oxycodone, and it is used for diarrhea, but now used in cancer. Itraconazole, it is acts as antifungal drug, approved for antifungal drug, but now it is used in prostate cancer. The best known example is aspirin, which is used as an NSAID, but now it is used in colon cancer. So these are the, I have taken the some example, but there were huge examples of drug repurposing. And the best is of COVID-19. In COVID-19 era, it is a very tough era for all of us. So no specific treatment is available for coronavirus. So many drugs were repurposed, like Ganovo, hepatitis C virus protease inhibitor, lopinavir, galidesivir. It is a RNA polymerase inhibitor which is used in yellow fever. And example is favipiravir, which blocks RNA polymerase. It is basically used as a flu drug, but in corona time it is used as an anti-COVID drug. And the best example we all know is hydroxychloroquine, which is anti-malarial drug. And in combination with azithromycin, which is an antibiotic drug, it is used in COVID-19 treatment. So repurposing have many advantages. Now, if we talk about the strategy of drug repurposing, it has two strategies: on target and off target. If we talk about on target DR, so in on target, the name itself indicates the known pharmacological mechanism of a drug is applied to a new therapeutic indication. Means the biological target of the drug molecule is same, but the disease is different. In this diagram, it is shown that drug target is known therapeutic indication which is approved by the FDA, and the new therapeutic indication which is done by the repositioning. The best example is minoxidil. Minoxidil is an antihypertensive vasodilator. Now it is transformed from antihypertensive vasodilator to anti-hair loss drug. How? As an antihypertensive vasodilator, minoxidil has the property to widening blood vessels and opening potassium channels. And this allows the more oxygen, more blood, and more nutrients to the hair follicles. And this pharmacological action helps its use in the treatment of baldness, androgenic alopecia. So the target is same. The target is known, but new therapeutic indication. If we talk about the off-target DR, so the pharmacological mechanism is unknown, and the drug acts on a new target for the new therapeutic indication. So both the target and the indication are new, which is shown in this diagram, like drug, known target, known therapeutic indication, new target, new therapeutic indication, which is done by the repositioning. The best example is aspirin. Aspirin is an NSAID, we all know. It is used in the treatment of various pain and inflammatory disorders. So target is known and therapeutic indication is known. It also suppresses blood coagulation like clot formation and use as an antiplatelet drug. Okay. But the new target is what it is used in now. It is used in the treatment of heart attack and stroke due to its antiplatelet property. And another use of aspirin is in the treatment of prostate cancer has also been reported. So In silico approach has gained wide popularity with significant success in drug discovery program. It has more advantage over the experimental based approach because it reduces the time and cost and development of the drug. But it has certain limitation also. The limitation of this method is that it requires the structural information about the drug. If the protein target is not available, then the disease specific phenotypic or genotypic information of drugs are required. The discovery scientists and researchers have combined in silico and experimental approaches to identify new therapeutic indication for existing drug, which is called mixed approach. There were certain mixed approach also. In the mixed approach, the result of computational methods is validated by preclinical biological experiment in clinical studies. So researchers combine both the methods. Okay, so this is the approaches of drug repurposing: experimental based approach and in silico approach, and the third approach is also there, which is called as mixed approach. If we talk about the experimental based approach, then the experimental based approach is also known as activity based approach, which refers to the screening of original drugs for the new pharmaceutical indication, but with the help of the experimental assays. Like it involves protein target based and cell organism based screens, in vitro and in vivo disease models, but it doesn't require any structural information. And several approaches are used, like target screening approach, cell assay approach, animal model approach, and clinical approach. But the main thing of experiment based approach is that it doesn't require any structural information of the protein or the target. If we talk about the in silico based approach, then in silico repositioning is done by the virtual screening of public database of huge chemical libraries. Like we have many libraries of the chemical structure or the ligands. By using computational biology and bioinformatics, chem informatic tools, we can go for in silico based approach. The identification of in this approach, the identification of the potential bioactive molecules, or we can say the phytoconstituent or the active molecule, is achieved. It is based upon the molecular interaction between the drug molecule and protein target, or the affinity of the drug molecule with the protein target. So there were the third approach, that is mixed approach, is also there, which is the combination of experimental based approach and in silico based approach. So combination is what? In mixed approach, the result of computational method is validated by preclinical biological experiment, like in vivo, in vitro tests are performed, and the clinical studies were performed, and they are combined with the in silico studies. So it is a combination of the molecular docking studies or the in silico studies with the in vivo, in vitro, and clinical studies. It is known as mixed approach. Now, methodologies of drug repurposing. So there were basically three methodology of drug repurposing. First is drug oriented, second is target oriented, and the third one is disease therapy oriented. There are the three approaches of drug repurposing. So if we talk about the drug oriented methodology, then it is based on the structural characteristics of the drug molecule, because it is drug oriented. Biological activities of drug molecules, adverse effect and toxicity, these all things are evaluated. It is meant for identify molecules with biological effect based on the animal assay or the cell assay procedures. It is based on the traditional pharmacology and drug discovery principles. in which we determine the biological efficacy of drug molecules without really knowing the targets. Target is unknown and the biological efficacy is determined. So it is a drug oriented methodology. Now, if we talk about the target based methodology, then in this it comprises in silico screening because the name is target based. So it comprises of in silico screening, or we can say virtual high throughput screening of drugs or compounds from drug libraries or the databases which are available, like ligand based screening or molecular docking. And this is followed by the in vitro and in vivo high throughput or high content screening, or we can say in vitro and in vivo assays of drugs against the target molecule. In this method, there is significant success rate in drug discovery as compared to drug oriented method. So next is disease therapy oriented methodology. This is different. In disease, it is included both disease and therapy oriented. So the application of disease therapy oriented methodology is relevant when there is more information on the disease model is available. And the drug repurposing can be guided by the disease treatment, which is based on the availability of the information or the availability of data. So data is given from by proteomics, disease specific target protein, or from genomics, yeah, metabolomics, yeah, phenotypic data like uh, off target mechanism, pharmacological target, disease pathway, pathological conditions. If we have all this availability of information, then we can go for this methodology. From all this, I had taken the in silico methodology of molecular docking technique I am showing here. I have done the repurposing of drugs for coronavirus also. So first we have to understand what is molecular docking techniques. It has some steps if we talk about the in silico technique. So first thing is selection of protein. First we have to select the protein. Protein is what? Enzyme. If we want to do the repurposing of drugs for diabetes, then we have to select like alpha amylase or alpha glucosidase, DPP4 or aldose reductase, whatever enzyme we want to take. Then identification and validation of enzyme from the protein data bank, we have to take the one PDB ID of that enzyme and check its properties. Then preparation of enzyme by removal of water molecule, cofactor and the binded ligands. Protein surface will creation of protein surface, and then last one is cavity detection work. So this is all about the selection of protein. If we talk about the selection of the ligand, so if we have a protein, then we have a ligand, like we have drugs, we have a phytoconstituent, we have a bioactive molecules. So first energy minimization of ligand is done, then the preparation of ligand is done. The next, after the selection of the protein and selection of the ligand, the next, the most important criteria is selection of the software. On which software we want to do the molecular docking studies or the reverse molecular docking studies. So there were paid softwares were available, trial version available, open access softwares available. We can choose anyone from this. And the last step is the docking process. Software is confirmed, then ligand and enzyme were import on the software. XYZ grid generation is done and docking wizard is open, in that the interaction studies were calculated. So I had given one example of coronavirus main protease 3CL Pro. I had chosen the 3CL Pro as my inhibitor, and my PDB is 6W63. It is a coronavirus which is in co-crystalline inhibitor X77 combined with X77 co-crystalline inhibitor, and its resolution is 2.10 Armstrong. So this is the raw enzyme. First removal of water molecules, cofactor and ligands has done. Then the surface is protein surface is created. See, this is raw and the protein surface was created. And the green portion is the cavity. So detection of cavity is done. I detect the three cavities and the protein is ready for the docking study. So the example is of 6W63, which is for the coronavirus inhibition. Now the second point is ligand. I'm working on this. So I had this is my ligand, that is RS321. It is an example, and this is the standard X77, which is covalently bounded with the 6W63. So I had taken this as a standard. Now, if we want a ligand, then we can download from data bank or FDA drug bank, zinc 15, chem spider, pubchem, Wolper, or we can draw in the camera also. But I had taken from the pubchem. Then the next step is minimization, energy minimization. I had done this on the cam draw, MM2 forceful is there. Then the preparation of ligand is done. Now selection of software. We have to select the software also. Many softwares are available. Cold, Schrodinger, Glide, Moligro. Autodoc Vina is free. Moligro, Sanjeevani. I was done my work on the Moligro virtual docker software. So this is the docking process. Import of ligand on prepared enzyme. First, the first thing is that we have to open our software worksheet and then import the enzyme and then import the ligand. Then grid generation like XYZ axis were confirmed, and docking wizard is open and interaction studies calculated. This is the means good examples like electrostatic interaction. This picture is of electrostatic interaction. This is static interaction. It is the hydrogen bond interaction. Blue lines indicates the hydrogen bonding and of my ligand with the protein. Now these are the results. See, in from the table, it was concluded that the my drug RX321 have Morlock score of -225.175, and the standard X77 have -156.913. So from the table, it was concluded that the my drug is have more affinity with the enzyme. It is binded tightly and potentially inhibit the enzyme than the standard drug. And this red color indicates the interaction of amino acid of the enzyme with my ligand, and these blue dotted lines are the hydrogen bond interaction. So I had repurposed the many drugs for coronavirus, and this is one example, and this is the example of the in silico approach. But recently I had done the mixed approach also for the aldose directed inhibitory activity for cataractogenesis, in which I had done the in silico approach, and it is validated from the experimental approach. So if we talk about the challenges of this drug repurposing, it has challenges also. So drug repurposing is a complex process involving multiple factors such as technology, commercial models, patent investment, and market demands also. So, some multiple challenges include choosing the right therapeutic area of the drug under investigation, is issue related to the clinical trials also, such as need to run new trials from start if the data from the clinical or the preclinical trials of the original drug is outdated or not satisfactory. So these are the some challenges. So in the brief, it was concluded that the drug repurposing have advantages, that it decrease the drug development time, reduces cost, and reduces the high risk of failure. So brief conclusion is the introduction of drug repurposing was there. Drug repurposing versus traditional drug discovery. strategies of drug repurposing, approaches of drug repurposing, and in the method I had shown the molecular docking studies. Thank you.
Thank you, ma'am, for your wonderful talk. You have covered all aspect of drug repurposing. Really, you started wonderfully with telling with various case studies and in introduction all the reprocessing, repurposing, retasking, reprofiling, and with all the pharmacological interactions, pro-drugs, and what are the various processes which includes time and risk of failure and the cost, pharmacoeconomics, all the regulatory aspects, preclinical, clinical, paraclinical, post-clinical aspects, and various steps of repurposing, drug repurposing, and what time it takes in traditional drug discovery and traditional repurposing, and various examples, live case study examples like of antidiabetic thiazolidine grips, rosiglitazone or oxycodone, itraconazole, aspirin, as well as at the time of pandemic COVID-19, a lot of drugs were repurposed. So beautifully you presented that too also, madam. And as well as you told one case study of minoxidil, potent vasodilator, which adverse effect was hirudism, and later on it became used in alopecia. And
Hmm.
off-target drug repurposing with the example of aspirin and various approaches, experiment based, and a lot of things you have told. So once a person from chemistry background watching this session can definitely come as an output of drug repurposing purpose. So thank you so much, madam, on behalf of ISF College of Pharmacy Internal Quality Assurance Cell under the MOU activity with Smriti College of Pharmaceutical and Education and Research. You have delivered this talk very precisely. So I am thankful to you, madam, for your precious presence. You have taken really your busy time out of your hectic schedule, busy schedule. So from all behalf of ISFCP family, once again, I am thankful to you, ma'am.
And I'm very thankful to you, sir, for giving me this opportunity. Thank you so
Thank you,
much.
ma'am. Thank you,
Thank you,
ma'am.
sir.
Thank you so
Have a
much.
nice day. Bye.